identifying Crohn's disease signal from variome analysis.
Hello, thank you for coming. My name is Yanran. I'm a PhD student at Rutgers University. I'm going to talk about identifying Crohn's disease signal via variome analysis. So first, a brief introduction of Crohn's disease. It's a chronic inflammatory disorder in the GI tract. It's one main category of inflammatory bowel disease. It often happens at ileum, but it can also happen at anywhere from mouth to anus. So biopsy is now often required for accurate diagnosis of the disease, and currently there's no cure for this disease. And it's also a very common disease. It's reported that Europe and North America has the highest prevalence and incidence. And up until now, there are a lot of GWAS studies about disease. Over 200 susceptibility loci have been found about the disease. And here is one result from one of the GWAS studies. You can see that on the x-axis is the odds ratio, and except for these 4 variant loci, the others are, they all have very low odds ratio and close to 1. So, and also you may know that it's really hard to reproduce one GWAS study to another. So our objective of the study is to use another method to try to find new potential causative CD genes and pathways, and we also try to build a predictor for predicting CD predisposition. So here is a brief flowchart of how we did this. So we take exome variant call files as an input, and we, in our pipeline, we generated the gene level functionality changes. We use feature selection to extract those genes, and we do further analysis for the features, the genes that we extracted. And we also utilize support vector machines to build models. We use cross-validation to validate our model, and further, we use a separately sequenced cohort to further validate our model. So to give you a clearer view of how we constructed the gene functionality changes, here is a protein coding gene example. So here, all the arrows are the variants covered by the exome sequencing. All of them, including the rare variants and also common variants found in those GWAS studies. We think all of the variants function, although all of the effects from the variant in terms of the function changes on the protein should be taken into consideration. So we summed up all the functionalities affected by the variants, and specifically for the other indel and stop and loss and gains, we give it a large constant, and for the synonymous ones, we give it a small constant, and for those non-synonymous ones, we we give it a snap scores, which is a prediction tool that give you a functionality predictions on the variant. So we also give it a factor for the heterozygous genotype, and we're summing all the scores into a gene score. So here our data becomes like this. So each subject is represented as a vector of different gene functionality changes, and we have the labels as CD and healthy. So we did a live one out cross-validation on the training data. So basically, each of the individual is taken as a test individual, and the other ones are training individuals in each of the procedure. And we validated the prediction results. And of course, we didn't use all of the gene features since there are a lot. So all available genes are negative control of our method. And we tried different external resources to help us build a prior knowledge of the data. So for the mesh genes, we did a natural language processing in the PubMed literature. That in the abstract, it says this gene is associated with Crohn's disease. And also for the GWAS genes, we took the genes that are in linkage disequilibrium with the GWAS loci of the disease. And we also went to SwissProt to extract the 18 genes that are reported in SwissProt to be associated with Crohn's disease. So in the figure, it's the result. The y-axis is the performance AUC, and the x-axis is the different random numbers of the genes that we choose from the different sets. And we can see that the external set of genes do not carry any CD signal at all. So we're pretty upset about this. So, but we did further analysis. This time we took help from a tool called Pascal. It is a statistical tool that took the previous GWAS statistics, and it translates those GWAS SNP statistics into a gene p-value. And this time we have a Pascal-ranked GWAS genes, and it's about 400 genes. So we did the same thing, except this time we took the top-ranked different numbers of Pascal-ranked GWAS genes, and it turned out it performed pretty well, especially for the top-ranked 175 Pascal genes. So we're pretty happy about this. It proves that our method works. But further, we were trying to find better performance, maybe even than that. So we did several feature selection methods. In the interest of time, I'm going to focus on one of this. It's a method it's a modified C45 method. And just to note that in order to avoid the biases, we also did the feature selection on the training part. So we found that the red dots and lines represents the DKM, which is a feature selection from our method. Then we find that the top-ranked 125 DKM genes has the highest performance, even better than the orange lines, which is the Pascal GWAS genes. So we're pretty happy about this. We chose this set as the genes for our predictive model. And here is how the prediction looks like from the cross-validation. And we just simply choose a cutoff between in the middle. So above the cutoff, we call it CD, and below the cutoff, we call it healthy. So, but in terms of a clinical diagnosis situation, I think it's better to tweak the cutoff in order to achieve better precision on the risk of losing some recall, or achieve better recall on the risk of losing some precision. But here you might think that it's only cross-validation, it should be biased. So I take another separately sequenced cohort. I'm gonna skip the details of how I remove the batch effect. And anyway, this is the result for the prediction on the model built on the training set and prediction on another set. So this is the performance. Again, we just use the default cutoff here. And in terms of a machine learning model that is built on a relatively small training set, I think it's a decent performance. So what are the model genes that allow for this performance? So we went back to check the genes that we found. Apparently, we found the genes that are known to be involved in CD pathogenesis, including the most infamous one, NOT2. And also, we checked if those genes are enriched significantly in the pathways, and it turned out they are. The most important, the interesting findings that we found, all the black labels are the genes found by feature selection, and these two magenta transcription factors regulates the expression of these genes simultaneously. And interestingly, this transcription factor is reported to be involved in CD pathogenesis. So we highly suspect that this one, now it's not reported in any literature, may be involved in CD pathogenesis. So lastly, we think, as Stephen mentioned in the morning, this performance is far from the clinical diagnosis, but I think with a larger data set and including more information, including such as gut microbiome, where sometime we will be able to achieve better performances in order to help clinical diagnosis. And we think the pipeline could also be applied for other complex diseases. And the next step, I'm going to get larger data set and also including more information in the gene score, such as if regulatory variants. So with that, I would like to thank you for all of your attention and these findings, and the members from my lab and members from our university and also our collaborators. Thank you.
Thank you, Yanran. Perhaps I missed it, but your model includes also counts the synonymous mutations. So
yes.
It includes a score for synonymous mutation.
Yes.
What is the rationale for that, and how would the model perform if you didn't include the score for synonymous, if you excluded synonymous mutations?
Oh, okay. So for the score scheme, actually, I tried a lot of different ways to construct the gene scores. Mm-hmm. And actually, this one is the highest performance that we can achieve. So the rationale for including the non-synonymous variants is that actually synonymous ones are the actually synonymous ones do affect the protein functionality. And this time, because the equation is heuristic, we only give it a constant. But I think in the future, when the predictions for synonymous effect on protein function is like have a higher accuracy, it's better to include that as well.
Okay,
Thank you.
Thank you. I think in the interest of time, we'll move on to the next speaker.